such an honor to be here. And first thing, I have to echo Nicole in thanking Mr. Schwab. It's the first time that I can give a talk with the title of the endowed professor, Charles Schwab Endowed Professor. So, and thank you for the opportunity to let me work on our common mission. I wanted to spend a couple of minutes talking about how the UCSF Dyslexia Center was born and how it's really kind of modeling itself to the success of the Memory and Aging Center at UCSF. So the Memory and Aging Center is a hub that is being built in the past 20 years at UCSF for the study of aging, cognitive disorders in aging. And it really takes the strength of the medical world to advance the cause and diminish the stigma of aging. And it really is a multidisciplinary team that puts together clinicians, scientists, social workers, nurses, community advocates, all together for the same goal. So a few years ago, when my own daughter was identified with dyslexia, I looked around and I said, well, where is a place like the Memory and Aging Center for her? Why isn't anybody in the medical world or in a multidisciplinary way helping me figure this out? If I can't figure it out, and I'm a behavioral neurologist, I have all these titles, and how can anybody else figure it out? And so right at that time, two visionary parents from the Charles Armstrong School, Steve Carnevale and Dave Evans, happened to come at UCSF and say, we want a better care and better understanding of our children who are Charles Armstrong, and also visionary educator like Claudia Kuczek. And so the whole thing came together and said, we have to do this. We have to build a center that really protects, takes care, does research, advocate for our children, because there is not a single person that can do it. There is not a single person that knows about the brain, that knows about school, that knows about education methods. We all have to do it together. So this was the story. We're not quite as big as the Memory and Aging Center yet, but with the generous help of philanthropists like Mr. Schwab, we're getting there. So, of course, at the mission of the Memory and Aging Center, we had to add a piece, which is the educational and school piece. And we were lucky to be able to work with excellent schools, starting with the Charles Armstrong schools and other schools in the Bay Area, like Chartwell and Athena Academy, that are already hubs where children with learning differences and neurodiversity are taken care of. So the idea is that we have a center in which we do research, we do medical care. I think these children have fallen through the crack, and adults and aging individuals with neurodiversity have fallen through the cracks. Is it an education problem? Is it a workplace problem? Is it a university problem? And the medical world has not been behind them. And in our society, the medical world is very powerful. We need it. We need to take care of families and children and adults without the need of spending thousands of dollars for private evaluation. So we have the medical care part, and then the partnership with the schools, and also being in the Bay Area, we need to take advantage of the incredible technology expertise so that we can better identify and better help children and adults with specific cognitive or behavioral challenges. So where do these behavioral challenges come from? I'm a behavioral neurologist, which is a little bit of a hybrid between a neurologist and a psychiatrist. So we really study the output of the brain, which is cognition and behavior. So we think of human behavior as an emerging properties of different networks in the brain. And we can think of networks like muscles. So there's different pathways that do different things. A big distinction that we can make to start with is left hemisphere and right hemisphere. We think that the left hemisphere is very involved in linguistic processes, but also processes that involve rote memory, memorizing things. The right hemisphere is the hemisphere more involved in visual-spatial functioning and in social-emotional functioning. This is something that is a little bit newer. We all kind of used to thinking of language and memory and visual-spatial functioning coming from the brain, but social-emotional reactions and abilities also come from specific muscles of the brain that we need to look at. So when Mr. Schwab says, I was a charming boy, that means he also has a strength in social-emotional functioning. It's not just something that we do acquire, but is also an intrinsic strength that comes from specific brain networks. So the idea of a whole brain assessment in dyslexia, but also for other learning differences, is that we need to look at all these aspects because they work in synergy. These different networks in the brain, and I'll show you some evidence of it, we now understand more and more that they work in synchrony one with the other. So if one is working differently, the other probably will too, and it might be the basis of some of the strengths that we see. So here is an example, really an analysis that we just finished with one of our neuroimaging engineer colleagues that really shows you this concept. So red means areas of the brain that are activated, and we don't need to go into details in what they do, and blue are the ones that are deactivated. So you can see that there is a balance. When certain areas are activated, others need to be deactivated. And for the red ones to activate optimally, there needs to be a downside in the blue ones. So there is really a balance that we're just starting to understand. So we don't think that the strengths of the dyslexic brain come just from the fact that, well, I couldn't read fast, so I need to look at pictures. No, the brain is probably, our theory is wired from the beginning to have a strength in this other function, for instance, visual-spatial functioning. So at some point, I think we will be able to identify slow readers even from their specific strengths. So going back to our networks, one of the let's say founding principles of our research is that we can have these predictable patterns of strengths and weaknesses. And that dyslexia is not a unitary concept, that here also comes from the pioneering work of Marianne Wolf, who you'll hear in a little bit, is that reading slowly is a symptom. So if this left hemisphere network is, for some reason, in development or in aging, which are our two most vulnerable times for our brain, functioning differently, you'll have very common symptoms. So in aging, the common symptom is not finding words, having difficulty finding words. In children, while in school, the most common symptom is reading slowly. But then we have to go deeper. We have to understand where it comes from which of those errors, pathways, or areas is coming from. Because if we don't, we're not going to be able to help the child or the adult in a specific way. And we've done this in the aging world by looking at anomia, so word finding problems, and dissecting it and trying to find which are the cognitive and neural bases of it. We were able to identify different disorders of aging that before we weren't able to identify, and we're very close to targeted treatments because of that. So this is the hypothesis also in the dyslexic neurodiversity brain. We really need to understand where the symptom is coming from. Kind of like if someone has a fever, a child has a fever, we wouldn't stop and just give them paracetamol. We just want to know where that fever is coming from, then we can treat it properly or help it properly. So if we think about all these networks in balance one of each other, we can start making sense of some of these terms and comorbidities that we've all went through with our children or with ourselves. And do I have this? Do I have that? Do I have comorbid ADHD or comorbid dyscalculia? And we can start thinking it from a system neuroscience perspective. So we can think that dyslexia can come from different cognitive mechanisms. So someone might have difficulty remembering words fast, or someone might have difficulties processing sounds, or having a visual or understanding difficulty, or an executive difficulty, and sensory motor. Maybe many of our children have difficulties with fine motor processing, and all of these different mechanisms can cause a slow reading. But we need to identify which mechanism is the cause of the slow reading. And then because of how those brain networks are organized, as you saw in the previous picture, which ones work in balance with each other, which ones are close to each other, then we can start understanding other functions, like social-emotional, which is in the right hemisphere, very near the visual semantic processing. And math. Math is a whole other domain, kind of like language. Is it high-level math, or is it math facts? And we can start looking at this as a neurodiversity spectrum in which there are different symptoms that most often than others can co-occur with each other. But then, if we think of the way that these networks and functions are in balance with each other, we can actually start thinking of how they are associated with specific strengths. So a child or a person who has a rote memory weakness for words will likely have a strength in the network that is in balance with that network, which likely, and we'll see some examples, would be visual strengths or semantic strengths, so understanding patterns or getting meanings, understanding meaning and problem solving in a way that is different and more and faster or more efficient than other people. Or another example, if there is a fine motor weakness, then we could predict that the network imbalance might be the one in social emotional processing. So we can predict that maybe children and adults who are not, who have dysgraphia or dyspraxia, these other terms that I've used, might be very strong in social emotional and need to be valued and protected because of that. So let's look at some specific examples. This is an example of a child that we saw at the Dyslexia Center. Oh, and there is a pointer. Great, thank you. Who had a very small, oh, now I need to choose where to point. So here, whoa, that doesn't get that far. So anyway, you can see the brain in the middle is the brain of this child that we were studying, and you can see that little red dot, which is exactly where auditory processing happens. So this child had difficulty with auditory processing, and you can, in the right schematics, you can see a smaller blue dot in that same area, and we can expect that the same network in the right hemisphere might be enhanced, and so he might have strength with visual attention and emotional processing. So from this approach, we realized that there were different phenotypes of dyslexia with different strengths and weaknesses. These are 3 children, all from the Charles Armstrong School, all diagnosed with dyslexia. We don't need to go through the details. We can look in the patterns of visual that the 3 children, each of different colors, are very different. They're very, they all 3 very bright. They all have very high nonverbal reasoning, but the patterns of strength and weaknesses in those networks is very different. Their anatomy is also very different. We have this technology now that we can look at single brains. This is a technique that is being used and that we borrowed from neurosurgery. So people that have to go into surgery to get tumor resected in the brain, the surgeons really want to know where the pathways are, especially the ones involved in language, so that they don't touch them. And so we borrowed some of the engineers and neuroimagers from that field to look at exactly how the pathways are organized in each individual child. And you can see that the symmetry between right and left of the brain of some of these pathways, you can see the blue one or the dark green one, are kind of different and almost opposite in a case. So the one in the middle, in the high middle, the green pathway is much bigger on the left, on the right, and in the one in the lower right, for example, the green pathway is much bigger on the right than on the left. So very, very different. And at this point, treated in a very protective, wonderful school like Charles Armstrong, still treated in the same way, still remediated in the same way, and for one child is working and for the other one is not. And that was basically the reason why Stephen and Dave came to us at UCSF to start with. It's like, why is the approach working for some kids and not for others? So let's look at some strengths that we are looking at. So we do these evaluation. Many of you have had children that came through the program. It's a long evaluation that we know when we need to, is kind of our discovery cohort that then through technology we hope will make the assessment easier. But now is long, is about 20 hours. It involves imaging, genetics, psychiatry, neuropsychology, emotion, visual-spatial, evaluation of all aspects of the whole brain. Some happens at UCSF, some happens in the school, but it's a long commitment, but everybody seems to be happy and enjoying it. So we'll look at 4 strengths. The first one that I'm really proud of, because it's very, very new and is very dear to my heart, especially because I think many of these children are not protected enough, and they have a gift of empathy, a gift of emotional processing that we have not previously recognized. So as we said, the left hemisphere is more involved in language, the right hemisphere is more involved in emotion processing. So the hypothesis is if these two, some of the networks within these 2 hemispheres are in balance, reading and language difficulties might be associated with emotional enhancement. So just as the children might have difficulty recognizing words because of a left hemisphere difference, they might have enhanced ability to recognize facial expressions and to produce facial expressions because of the balance between these 2 systems in the brain. So I'm a behavioral neurologist. I study language. That is my expertise. I was lucky enough that in a place like UCSF, there is the expert in emotion physiology measuring. And so I could just convince her, Virginia Sturm, and say, hey, we need to look at this, and she could take all her tools and her lab and concentrate the efforts on seeing also of our children. And this is very important kind of bridge between psychiatry and system-based neurology, because we for the first time are able to measure emotions, not just ask parents, well-meaning but anxious parents about their children. We're actually looking at the children's response. So children are in this lab, in this room at UCSF. They watch videos that have been standardized and coded. We measure heart rate, respiration rate, skin conductance, basically all these measures in the body that produce really emotions. So they are hooked up, and this is an example. Many of you have seen this video. So the child is looking at the video, and we measure her facial expression and her heart rate and respiration moment by moment as she's watching that video. It's almost impossible, right, not to react. And actually, one of the things that we ask the children, we look at emotional regulation, we ask them not to express also their feelings while they watch the videos. So the finding that is really consistent, and is probably going to be, is our first, second paper from the center, is that there is enhanced emotional reactivity and understanding in children with dyslexia and the phonological classic type of dyslexia. So this is a strength. There's going to be a strength for them in their life, but it also puts them at risk, especially in the critical times in their life. So from young age to adulthood and in aging, we need to protect them, because understanding emotions more means that we're going to react to them more. So this is an example of a reaction to disgust, and you can see the difference of the child on the left. This is a disgusting video, and the child on the right. It's a pretty disgusting video cleaning earwax from it. So they can't get more different than this, and it is really something that we want to look at. Okay, And now we have brain images from all of these children. And so we say, oh, is this just like a random fluke that you're finding? No, because we see actually brain correlates of it. So there's changes in the dyslexic brain. I won't get into many details, but in the right hemisphere regions that are involved in emotional understanding and emotional regulation. So really important point that we shouldn't just look at language in these children. We should really take this whole brain approach. So another skill, visual-spatial memory. This is the task that we use in visual-spatial. Very different from the classic task that is used in the educational, usually academic batteries, in which children are asked to copy figures. Here they're playing this video game in which they're navigating around a virtual environment. It's a very well studied task. We even know how mice do this. We know which genes regulate this task. We know which neural networks are associated with it. So they need to learn 15 turns in this virtual environment. And most of them use a strategy that is called allocentric, so they take landmarks from this virtual neighborhood. It's really hard. It's 15 turns that they need to memorize. And then they have to get it right twice in a row. And then we ask them again after 45 minutes. What I really like about it is it doesn't involve fine motor movements. So it kind of dissociates visual memory from fine motor skills, which are necessary to copy figures. So how do they do? Not all kids with dyslexia do extremely well, but there is definitely a subgroup. You can see them circled there. The distribution is different. Out of 30 kids with dyslexia, there are 9 who are exceptional. So while in the general neurotypical population, there are 14% of super visual learners. This is really visual memory. In the dyslexic cohort, there is 30%. And is not correlated to general measure of visual IQ. So what I think this indicates is that it's something different. It's not just that they're smarter visual spatially, they're doing this task in a different way, probably because of this difference in the wiring and in the synchronous functioning of their brain networks. Furthermore, we would have missed them. If we just did the copying of figures in the classic neuropsych testing, we would have missed them. It doesn't correlate. So one can do really poorly in the classic tests of copying figures and remembering them after a few minutes, and still do extremely well at this task. How about in the brain? In the children who have this superior visual memory, we definitely see bigger structures, more connected structures in the right hemisphere. So you can see in the panel at the top, on the right, there is this structure that is called the superior longitudinal fasciculus on the right, and the same structure on the left, and you can see visually that is thicker in the right on the left. So again, there is a neural correlate of that. Another very important function that we've talked about before is semantic conceptual abilities. Again, there is a spectrum, but there is a subgroup of children that are really superior in finding pattern and grouping information based on their meaning. And these are usually the kids who have a rote memory difficulty. So if someone has difficulty in remembering things by rote, the way to remember it better is to make stories out of it, to see patterns. And then that becomes a problem solving and a creative thinking ability. So not everybody, but again, there is a subset of dyslexic children that are really good at this. And in our cohort so far, it's still small. We need more to make these more robust finding, but it's a different subset from the visual-spatial super learner. So it's not, there is this association that is not always so easy, so we need to look at the individuals to really identify it. Again, we see a neurocorrelates. This other pathway is called the arcuate fasciculus in the semantic learners, which is a name, the temporary name that we're using is much thicker on the left than on the right. So there is a neural signature associated with this strength in these children. Lastly, and I thought this was very interesting, it's very preliminary, but I thought it was very interesting today talking about entrepreneurship. So there is a field of neuroscience and neurology that is called neuroeconomics, and studies the way that people make decisions and how they deal with uncertainty. So many economic decisions are made in the face of uncertainty. And so there is a whole field of neuroscience that studies how people make these decisions and how they deal with uncertainty. Now, we all know from our own experience or our children's experience that children with dyslexia deal with uncertainty every day. They look at a word and they just try. They don't know. They are uncertain on how they're gonna approach it and what the result is gonna be. So Krista Watson in the center is an expert in neuroeconomics, and she used a task to look at this in our children and see how they react to uncertainty. So is this a strength? The way they make decisions, is it a strength that could have to do with entrepreneurship? And and understanding how children make decisions, children with dyslexia make decisions, can it help us from the school point of view and the remediation point of view? Can it help us create better strategies for learning for them? So this is the task. It's a very well-established task that, of course, has never been used in children with neurodiversity. So they have to fish, and what they know is that they have several trials, but every red fish is 5 points, but if they catch the yellow fish, they lose everything. And there is so there is different amount of yellow fish in the tank, but they're uncertain because, let's see if I can play it again. Because at some point, the algorithm actually makes them get a yellow fish. So basically, what the task does is it changes the probability of them getting a yellow fish and losing everything. And the children need to learn whether for that trial, they're okay with the amount of points they have collected, or they want to take the extra risk and go another time. So there is a very specific cognitive model, and the task is constructed in a very precise way with Bayesian probabilities that we don't need to go into. But the idea is that there is two main concepts that we can look in this task. This is how children assess risk and how consistent they are. So to give an example is this of what children say during this task. For the first construct is, this time I'm going to catch the yellow fish, I just know it. And in the second one is, well, I got 5 points already, but I want to see what happens if I fish again. So consistency means basically how high they put the bar for themselves. And these are our preliminary results showing that really children with dyslexia have different decision making. And this is, again, the phonological, more classic type of phenotype of dyslexia. And what they do is that they set a bar very high, and they tolerate risk. They have higher tolerance for risk. And so I will let the entrepreneurs in the next speakers think about this and how this relates to entrepreneurship. So in summary, I hope I convince you that a whole brain approach is a novel and very relevant approach to look at children with neurodiversity, with dyslexia, but also other learning differences. That is not enough to just look at how they spell and they read, that we need to get an idea of their strengths and weaknesses. There are many strengths that can lead to many successful careers in their life, and hopefully we'll work with Nicole to figure that out. That we are more concentrated in the K-12 and in the aging cohort. So we have the whole age span here between the two institutions. We think that interventions should include knowledge about the strengths of the children, and that the approach should be having the right teaching method, but also specific cognitive training tools for the specific difficulties, and that's where the technology will help us. And then none of the different professions can solve this problem alone. We need to work together, clinicians, scientists, teachers, legislators. We have one of our first studies in a prison here in California to look at the prevalence of learning disabilities in the prison population. We know that we need to stop the downward spiral that we heard about before. So what is stopping us? What is stopping, and nothing is stopping us, but what is needed for the Dyslexia Center to match the impact of the Memory and Aging Center? And one frustration that I've had in starting this center that was fortunately was remediated by the generosity of Mr. Schwab and other donors is that NIH, the National Institute of Health, does not fund this research. And this goes back to our initial thought of these kids falling through the cracks. Is it a school problem? Or is it a medical problem? Or is it a scientific problem? And in all this question, they fall through the cracks. So look at the amount of funding that NIH gives to Alzheimer's disease or to aging disorders compared to dyslexia. We really need to work together on changing this. And although these are not diseases, we don't want to medicalize too much, we need to bring the medical and health world into the field. And this is what happened at the Memory and Aging Center. This is the funding scheme of the Memory and Aging Center. It started with a very generous donation as an endowed chair to Dr. Miller, who's the director of the center, and then both philanthropy and NIH funding started going together. So I hope that the future of the Dyslexia Center will depend on our generosity, but that we'll soon be able to also convince NIH that this is a problem that needs to be tackled. So I want to thank everybody for being here, the children and the families. Many of you have trusted us with your children. Thank you. It's an incredible honor. All our staff and collaborators, teachers, doctors, the psychologists, basic scientists, all working together for the same cause. And again, thank you to Mr. Schwab and our other generous supporters.